Hello friends, welcome to BMH Learning. SNP microarray known nucleotide sequences as probes to hybridize with the tested DNAs, allowing qualitative and quantitative SNP analysis through signal detection. SNP chips are DNA microarrays that test genetic variation at many hundreds of thousands of specific locations across the genome. Let's understand it using an example. Three persons' DNA is shown. Individuals 1 and 3 are homozygous for a particular sequence, while individual 2 is heterozygous for this sequence. Let's assume that the sequences we are interested in differ by a single nucleotide. In one sequence, a C exists in a particular position, and in the other sequence, the C is replaced by a T. This difference is nothing but a single nucleotide polymorphism or SNP. To identify the SNPs, the DNA is first copied using PCR. The DNA strands generated by PCR are separated and applied to the DNA chip. The short pieces of DNA sequences on the chip are complementary to the DNA containing the SNPs. Note here that the PCR products from the heterozygous individual differ at one position, either containing a C or a T. The short DNA sequences and just before the SNP we are talking about. If a DNA polymerase enzyme is added to the chip, it will incorporate a nucleotide that is complementary to the SNP into the end of the short strand. For experiments like these, the nucleotides to be incorporated are initially labeled with fluorescent color tags. In this heterozygous sample, a G has been incorporated into one strand. On the other hand, an A has been incorporated into other. So this leads us here. The heterozygous sample contains two colors, whereas the two homozygous samples each contain just one color. So, with the help of this technique, scientists can tell what SNPs a person possesses and whether the person is homozygous or heterozygous. This was all. Thanks for watching.